Okay, so we're going to move on to our next speaker, who's Dr. David Segal. David Segal comes to us from the University of California at Davis. He is a member also of our Fast Fire team, and his focus is on gene editing and research in treating neurologic disorders. But before he starts speaking, all I want everyone in the room to realize is that there's something incredibly special about Dr. Segal, and that is that he is actually, I know it's hard to imagine this, but he's actually an incredible athlete, if you didn't know. And Dr. Segal's not only an athlete, but he has the heart of a gem. He actually has run the China Great Wall Marathon. He has run various marathons throughout the country, of just one last week, and he's also flown with himself, his helmet, and his bicycle to Philadelphia to ride 77 miles in the $1 Million Bike Ride, all of which he did to raise money for FAST. I can't run a mile. I cannot run 1 mile, and this man has probably run well over 500 miles or more for FAST. So we can't thank you enough.
Well, thank you for that introduction. Wow. Yeah, that's fun. Okay, so today I'm going to tell you about CRISPR-Cas. So show of hands here, how many people in this room have heard of CRISPR? There's one guy over there who hasn't heard of it. Okay. So I'm going to tell you a little bit about CRISPR-Cas today, but I want to start off just by framing this as a kind of personal story, if you will. When I was thinking about what I wanted to do in biology in my life, and I went to graduate school. It was in the early 90s, and I was really excited about this idea of gene therapy. And gene therapy was just starting. And a little bit later, you're going to hear about gene therapy from one of the foremost pioneers in the field of gene therapy. So I'll leave the story to him. But I just wanted to say now that gene therapy is now a thing. So when I was just started graduate school, we were talking about gene therapy and what it could do, and it seemed like it made so much sense. And for people that had genetic mutations, they were going to put these healthy genes in a virus and use the virus to get the gene into the person, and that was going to be great. That's the way we're going to treat all genetic disease, and it all made sense, right? But in the beginning, it had a lot of trouble. It had a lot of trouble. And it looked like things were not going so well. And many people that were in the gene therapy field eventually left the gene therapy field, and it left what I would call the true believers. And the true believers stuck with it, and they met all those challenges. They figured out what the problems were. They figured out how to get around it. And today we have gene therapies. And so, this is a very interesting story. When we think about technology, people say that people tend to overestimate the value of new technologies in the short term and underestimate their value in the long term. And so when I tell you about CRISPR now, just try to keep in mind that right now we're kind of in this bubble phase that's still getting bigger, where we're sure that CRISPR is going to be the solution to every biological problem. Inevitably, it will not be the answer to every biological problem, and there's going to be some successes and some failures. But I think that over time, this is going to be something that's going to be extremely, extremely powerful. And I can say that because now we have gene therapies. And I teach a course in gene therapy at UC Davis. And every year up to now, I've been telling the students, go and do a report on what the progress is in gene therapy. Go and do the report on what they're doing in HIV or in eyes. And for the first time, I could teach the class, and I could say, tell me about the gene therapies that have been approved. It's really exciting. But in thinking about, so in thinking about gene editing and CRISPR and how this came to be, I'll just tell you a little bit about some background. So the idea that we could change the DNA in our cells or in any species cells is actually not a new idea. People have been doing this for a long time with trying to mutagenize plants or animals, worms, flies, something like that. They would use some kind of a mutagen chemical that would put some mutations in their DNA, and then you had to select for the organisms that had the color that you wanted or the phenotype that you wanted, some condition that you wanted, and then you could go back and study those genes. And even up to just a few years ago, these are still methods that have been used to manipulate plant genomes and model organisms. And that really was the way things were until about the mid-'80s, when people like Mario Capecchi, Oliver Smithies, and Evans came up with some methods that really allowed us to actually target a specific gene. And they were studying how cells go through this process of homologous recombination, which is something that all of our cells or all life forms, diploid life forms do. And in studying this, they realized that they could design a long piece of DNA in a certain way that when they put that into the cell at some very low frequency, as is shown in this diagram here. I don't know if you can see my cursor. But they could put in a very long piece of DNA into the cell, and it would align with the same kind of sequence on the DNA. And at some low frequency, a miracle would occur, and this little green box got incorporated in exactly the precise place that they wanted it to be. And that miracle occurred in about 1 in a million cells. But they were able to work with those low frequencies and select for the cells in which those correct events had occurred, and quite appropriately got a Nobel Prize for these sets of methodologies. So all the knockout mice that we've ever used is thanks to this kind of technology. And it has had incalculable benefit to human genetic research. However, it's instructive to consider that the similar methods didn't work for even something as similar as rat or plants. And in fact, outside of mice, there was no species that worked with this kind of gene editing approach. So we couldn't use it for therapy. And so many people started to look at this and they said, what is the slow step in this? Why is this only happening in 1 in a million cells? And basically, kind of in the mid-'90s, it was realized that if you could make a break in the DNA of the cell, and the chromosomes in the cell, that was the slow step. And if you could somehow do that, you could stimulate this process by hundreds of thousands of fold. Okay. Now, this was great because it opened up this possibility that we could actually go in and manipulate genetics in living cells and potentially living organisms. But back in those days, we didn't have a good way of making targeted double-strand breaks. So I was one of those guys in the mid-'90s that was working on this. And like I say, back then, every experiment was proof of concept because we didn't have a way to go and say, I want to cut this DNA sequence and just cut that. So everything was done with some natural large nucleases that could cut a very rare site in DNA. And if you could put that site into your experimental system, you could show that all this stuff would happen. But it kind of set off, I don't know if I call it a race, but it set off a kind of challenge to try to come up with ways that we could say, I'm going to target just this gene in this animal, and I'm just going to cut there, and then I'm going to do all these genetic events. So we're going to fast forward now to the modern day, and we have not one, but actually several technologies now for doing this. The first ones that come along were actually these zinc finger nucleases, which is something that I spent a lot of my career on. And there's another one called Tollins, which had its time for a few years. And then more recently, there's the Crispr system. And the Crispr system was really exciting to a lot of people, not because it necessarily did something that the other nucleases didn't do, but it was just so much more easier to use. It was so easy for almost any lab to do it, and then you could do these things at high throughput, and it had many advantages like that. But CRISPR is a targetable nuclease, like all the things I'm showing up here on that white panel. And all of these things can be used to now make targeted strand breaks in the DNA, which lead to all these downstream consequences. So one of the things that happens is if you make these targeted double-strand break in the DNA, the cell will try to repair that break. And it will do that in the most easiest way possible. And sometimes in doing it the easiest way possible, it makes mistakes. Not often, but enough that we can set it up so that if you just use this nuclease and you cut the DNA, you can start to make mutations at that point. And people have exploited this now to make mutations in specific genes. And so they could actually do this experiment in a cell and they'd say, I want to knock out the phytase gene or something. And now you can design this system so that you can just make mutations and stop the gene function from what it's doing. And then the other thing is if you make that double-strand break and you set it up with some kind of repair information that tells the cell how that should be repaired in a very precise way, the cells will sometimes use that information. And again, you'll get your green box inserted precisely in the genome where you want it. So this could be a gene correction. You could be changing a mutation into something that's no longer a mutation. You could be adding in something to the genome in a very precise way so that now you can put some kind of a tag on a protein or something like that. And so it's really, the field is explosively evolving all the time now. And certainly people could think about trying to use these approaches for therapy. So the first type of nuclease to enter into a clinical trial was actually a zinc finger nuclease, and that was used in HIV. Now, HIV is a disease where the virus gets into the blood and it infects the T cells, and the T cells can circulate all over the body. And so you'd say, how can you possibly treat a disease where the cells are all over the body? How do you get it to all over the body? But HIV has a kind of Achilles heel in that all the cells come from these blood cells, and if you can, get it, if you could somehow make those cells resistant to HIV, then the cells can't infect them. And so there is a gene called CCR5, and that's a gene that there's people that have natural mutations in that gene. So we think that if this gene was somehow, its function was knocked out, more or less the people are okay. There's not that many spare parts in the genome, but this seems like one where, let's say, there's redundant functions. So this one could be knocked out. But the virus, the HIV virus, needs this. So what happens is if you mutate this gene, essentially the virus will just bounce off the surface of the cell and not infect it. And in fact, people with this disease are naturally resistant to HIV as well. And so what this company, Sangamo Bioscience, did is they would take these cells out of patients that have HIV, because they're blood cells again. So they could take them out, they could treat them in a dish, and then they could put them back in. So this is an example of what we call ex vivo gene editing, where you take cells from the patient out, you can manipulate them in a dish where you can control a lot of things, and then you put them back into the patient. And this now went into clinical trial. So this was the first kind of nuclease application that went into clinical trial. And this is still in phase 2-3 clinical trials. But it is one of the most promising approaches to treating HIV because it seems to work. It seems that the cells are really protected from HIV. And since the HIV is killing the other cells, these cells have a selective advantage. And so that's a very exciting application in itself. Now, you could also use this, as I said, to put in a gene in a very precise way. And so in this case, this was also a trial that was done by Sangamo Bioscience. I should say I have no relationship with Sangamo Bioscience. I'm just using this as examples because this is the way the history went. In this case, they were trying to treat either lysosomal storage diseases or things like hemophilia. These are cases where you can have a soluble protein that goes around and has a beneficial effect. And what they were doing is they were using this nuclease, and they were cutting in a normal liver gene, a gene that's normally expressed at high amounts in the liver. Now, the liver is not the site that they need to correct for these genes, but what they did is they were able to insert a copy of their therapeutic gene. So let's say for hemophilia, they would put in factor IX. And they could put it precisely into the open reading frame of this highly expressed liver gene. Now that liver would express a high amount of this factor IX protein. And they could show that it went all over the body, and it actually, fixed the animals, and the hope is that we'll fix people like that as well. But to do this, okay, you have to take the nuclease and put it into the person, into their liver, right? You're not taking this out anymore. So these clinical trials started just last year, and the first in-person use of a targetable nuclease are these clinical trials, and that happened just last year. So the first in-person use of a nuclease just happened last year. So, just to say, it's like all happening right now, okay? So this is an example of what we might call in vivo somatic cell editing. Somatic meaning not the germline, not the cells that are going to go on for reproduction, but all the other kinds of cells like liver cells or brain cells. And there, you would deliver the nuclease. Often it's packaged into some kind of a virus or some other kind of delivery particle. And then you're putting the nuclease into the person. So now you can see this kind of ups the stakes a little bit, right? You can't control everything so well like you can in the culture dish. You can't select out the cells that got the right edit from the ones that got the wrong edits, if that's what's happening. And of course, if it does make something wrong, you can't go in and take those cells out, right? So this is getting to be more tricky. There was actually a CRISPR clinical trial for in-person use that was entering clinical trial, and it was halted due to concerns. But again, these are developing technologies, and people, lots of people, are very excited to work out these kinds of complications. Now, not only can we use these in therapy, but they're being used widely in research and for many, many other purposes. And because this technology now is so accessible, they're not just doing this kind of gene editing. Before I said the Kopecky type gene targeting technology only worked in mice. This works in every known form of life, from bacteria to humans. So this is just a list of some review that I wrote a long time ago. Now it would be silly to write a review like this because it's been used all over. But you could just see on this list, gene editing has been done. These are viruses. These are all plants. Sea urchin, I don't even really know how to say what that is. These are like fish, and these are mammals up here, like pigs, chicken, cattle. In many cases, the way that they would do this, like in mice here, or in rabbits, or in catfish, they just inject the nuclease right into the one-celled embryo. So after fertilization, they inject that nuclease right into the embryo. They can change the DNA in the one-celled stage, and then when you grow up that mouse, now all the cells of that mouse would have this genetic change. And this was going on. So for me, it was not a big surprise when I saw this paper, because people were editing embryos in dogs and cats and mice and rats, and then someone edited a human embryo. And I was like, well, yeah, it's DNA, it's a cell, of course you can edit it. But this paper set off shockwaves really around the globe, because when people really saw an example where you could edit the human genome, it really sent a wake-up call around that, seriously, we can start editing people. We could start designing the genetics of people. And this technology is it's here. So this was in 2015. And when this paper came out, there was a big response. So this is now germline, in vitro germline editing, where you take out zygotes, you edit the embryos, and then you reintroduce that back into a female and grow up a baby. And so when that paper came out, there were these big summits that were held about germline gene editing and what are the ethics surrounding germline gene editing. And they got together the National Academy of Science in the United States and the Chinese equivalent of that and the UK equivalent of that. And they were all held these meetings to discuss the ethics. And there were laws that were passed about whether you could use CRISPR or gene editing in general to edit embryos. Some countries banned it outright. Some just made restrictions, like the United States. And again, this is an emerging kind of field. But then there's a lot of pressure also to push this forward. And some people say, well, if you could do something to cure these genetic diseases, we want you to do it. We want this to happen fast. And then suddenly it happened. And it happened just literally a few days ago, that at the second summit on gene editing, a researcher claimed that he had edited embryos, human embryos, and had grown them up to become gene-edited babies. Now, this is a, I mean, not to be too dramatic about it, but this is a kind of a turning point in the ability of our species, because it's the first time that we not only understand genetics, but actually are starting to change our own genetics. And I would say that I think people would like to have this ability to try to fight debilitating diseases, but we certainly want to know everything we can about this, and we want to conduct this research in the most socially and ethically responsible way possible. And this guy did not do that. So this guy has been universally condemned, I would say, for his actions. And again, this has only happened a few days ago, and we're starting to see what the consequences of his actions are, because there are concerns about the technology, and like every other technology, it can be developed and it can be very beneficial, but we need to be careful. So with those things in mind, and the gene that he actually edited was this HIV, which I thought was, or the CCR5, which I thought was interesting. It's also the first gene therapy that went to clinical trial. So what might this mean for Angelman syndrome? So as the great Dr. Art Baudet had said earlier, it may be possible to use CRISPR to actually fix the mutations that are involved in UB3A mutations. I think there's some imprinting defects that are also caused by mutations or small deletions. And in principle, this kind of approach could be applied. Now, I don't know how easy that's going to be to do. And today, this is not something that I think one would want to run out and try to do. I think we need to know a lot more about the safety of this and how this might be used and what the consequences of this could be, and how we know that this is absolutely positively safe and effective. And today, we don't know that. But, like I say, there's many, many people that want to be able to use this to treat genetic diseases, and so this is something that many people will be working on. But so this is a possibility. Now, CRISPR could be used in some other ways to work for Angelman syndrome as well, and Mark Zilka is actually working on an approach like this. Now, I don't think his work is published, and I don't know a whole lot about what he's doing, but I would just say that he's using CRISPR-Cas to cut regions of the DNA, and he seems to have found some region of the DNA that he could essentially mutate, and when he does that, UB3A comes back on. So I think there's a lot, he's doing this, and his story will go on, and we look forward to reading about that. But it could be a very interesting use of CRISPR that is something that is, could eventually become a kind of therapy. Certainly, we want to know a lot more. Aside from cutting the DNA, it might be possible to cut the RNA with a different kind of CRISPR, maybe not CRISPR-Cas9, but some other variant of CRISPR to cut the RNA. And this could be an approach that's kind of like the antisense approach, where essentially you're not changing the DNA, but you're attacking that RNA, allowing the UB3A to be turned on again. So I think this is something that can also happen with the technology that we have. Again, we have to make sure that this approach is safe. There might be other things that we could do with this CRISPR technology, or one of the other technologies. And that would be to not cut the DNA and do all these genetic changes that I just said to you, but maybe something that's a little potentially safer might be to just change the information that controls how the genes are expressed. And so in the work that I've done and we've published, we took not Cas9, but a protein that's like that. It was a zinc finger protein, actually. And we used that to inactivate this, the gene that makes this very long transcript and allowed UB3A to come back on. And I think it should also be possible to use it in some other ways that we can make it so that by changing the epigenetic information, that information that controls which genes are expressed and which are not, we might also be able to use that in a way that just allows UB3A to be expressed in the brain again without making targeted double-strand breaks. And all of this, is, like I say, is happening in real time, but I think there's a tremendous potential to do treatments and potentially curative treatments in this disease. And so I'll stop there and take any questions. Thank you.
While we change over talks, do we have any questions in the room?
You mentioned Dr. Zilka's work. Are you aware of anyone else who's utilizing CRISPR to
For Angelman syndrome?
for AS, yeah.
No, I'm not aware of anyone else that's doing it. But there could be people in places that I don't even know about that are thinking to do that. The technology is that accessible. I expect that we will see other people starting to use it. But again, there's a difference between what we can do in cells today and what we would want to do for therapy, at least in my mind.
I'm nervous because I don't speak the scientific language very well, but my question about CRISPR in UBE3A mutation patients, what would be the downside to fixing those who have a stop codon, to fixing that simple thing? What could go wrong?
Well, let's put it this way. If the gene editing goes well and it fixes that stop codon, there's nothing that would go wrong with that. I mean, if we could do that, that would be great.
Right.
But usually, if we have a dish of cells and we treat those cells, not all of the cells will get that correction.
Okay. And then
and
putting
so-
that all back in.
Yeah, and remember, there's like 1 billion neurons in the brain.
Right.
And so, the question is, how efficient is this going to be? Are we going to do it in every cell? Is it going to cut in just the place where we want it to, or is it going to cut somewhere else? Is that break going to be processed in a way that we didn't predict? And, I think that, all these things are going to be tried, and they're going to go through animal models, and we're going to see it. And we're probably going to see it pretty soon.
So we still can't pinpoint where that cut is what you just said, right?
Well, we're getting much better at that. Yeah, we're getting much better at that. But like I say, I think today it's not ready to be used as a therapy, but many people are trying to make it ready to be a therapy, including many companies. So there's a lot invested in this.
Yes. Fingers crossed.
Fingers
Thank you.
crossed.
Great, thank you, Dr. Segal.